My name is Jayla and I was assigned KRAS. Glycine amino acid 12 is replaced by aspartic acid or cysteine, G12D or G12C. KRAS is found in the endoplasmal membrane of the cell, usually found in animals. By transmitting signals from the exterior of the cell to the nucleus, the KRAS protein functions as a molecular switch that regulates cell growth, maturation, and death. Normally, the KRAS gene acts as the hub for signals that promote cell division, but when there's a mutation in KRAS, it causes cells to multiply and send out excessive signals, which leads to cancer. The precise cause of KRAS mutation is unknown, though heredity might be involved, but KRAS mutations can happen spontaneously or as a result of exposures to environmental contaminants like radon or tobacco. Numerous cancer forms such as pancreatic and non-small cell lung cancer are linked to KRAS mutation. All human cells contain a gene called KRAS that serves as a key regulator of signaling pathways responsible for cell proliferation, differentiation, and survival. One single type of KRAS mutation called KRAS G12C accounts for about 44% of all KRAS mutations. G12C is a single point mutation with a glycine to cysteine substitution at codon 12. So for the normal function, the KRAS gene codes for production of the KRAS protein, which is a component of the RAS and MAPK signaling pathway. The protein transmits information from the cell nucleus from outside the cell. These signals tell the cell to either mature or take on specialized roles or grow and divide. KRAS is a GTP phase, which means it changes the GTP molecule into the GDP molecule. The GTP and the GDP molecule operate as switches that activate and deactivate the KRAS protein. It must bind to a GTP molecule in order to activate the transmission of signals, which means if the GTP molecule does not activate the KRAS, it will not send any signals. So this would be like the GTP, and it has to activate for it to send signals. So this is important. The KRAS gene is a member of the oncogenes gene class. The oncogenes can cause normal cells to develop into cancer when they are modified. This mutation makes the KRAS protein more active because it increases the signaling pathways leading to the oncogenes. KRAS itself doesn't have any subunits in the way a multi-subunit protein complex does, but its function is highly dependent on interactions with other proteins in its state of activation. This mutation causes a substitution of the glycine, which is the original amino acid, for cysteine and aspartic acid. So this will be the glycine, which is the original acid, and it will substitute with the cysteine or the aspartic acid. The KRAS is impacted by the G12C or the G12D because they prevent it from reaching the JTP and the JDP. In glycine, amino acid consists of carboxyl and amine groups and is hydrophobic and nonpolar, also neutral and uncharged. Glycine is the smallest amino acid compared to the other 19. Cysteine consists of carboxyl, amino, and sulfhydryl groups, which is hydrophilic and polar. So how does this affect the function? The mutation affects the protein by speeding up the growth, which causes cancer. For my protein, I use the saline pipe paper and the construction paper, also the pom-poms to create. Shows the unmutated, and this is the mutated, and this part shows the glycine, which we know that the glycine is one of the smaller amino acids, but when we show the mutated, we can see how this glycine changed. And then at the bottom, we can also see the GTP on the bottom left, which also is shown on the mutated one as well. We can see the GTP. Would be not being able to show all the amino acids due to it being a paper representation, and since it is a paper representation, it was hard to represent all of that, and then also it was hard to show the exact structure, but I tried my best.